haploid production. I told you the haploids from doubling JC, you'll get the seedless variety. Then you have the protoplast fusion. Then secondary metabolite production, cell suspension cultures of callus suspension cultures. Then you have the secondary metabolites. So many metabolites plants will be producing that you will be having. Then genetic engineering. Genetic engineering lopala, after you make the vector construction చేసిన తర్వాత you put that particular vector into the plant, either through agrobacterium or through the bioride gun. And how you develop the plants, you develop through the tissue culture method only you will be developing. So you have so many different applications. All these applications మనం నెక్స్ట్ వచ్చే క్లాసెస్ లోపల ఒక్కొక్కటి we will be studying in detail అన్నమాట. Next, first we'll come to the micropropagation. So micropropagation is nothing. The name indicates micro అంటే చిన్న దాని నుంచి propagation చేయడం అన్నమాట. అంటే ఎక్కువ propagation చేయడము. From small you can do the large type of propagation. దాన్ని మనము micropropagation అంటాం. For example, I will ask you a question. ఇప్పుడు we are taking seeds. Okay, you take ఒక వడ్ల గింజ అన్న తీసుకోండి, or you take చింత గింజ అన్న తీసుకోండి, or you take పల్లీలు గ్రౌండ్ నట్ గుడ్డలన్న తీసుకోండి. If you put one in the soil, in the suitable soil, how many plants we will get? ఒక్క వడ్ల గింజ నుంచి మనకి ఒకే ఒక రైస్ మొక్క వస్తుంది, ఒకే ఒక పీనట్ గ్రౌండ్ నట్ నుంచి ఒకే ఒక ప్లాంట్ వస్తుంది, ఒకటే ఒక చింతకాయ గింజతోటి ఒకే ఒక ప్లాంట్ వస్తుంది. బట్ here when you are using the tissue culture, what you have is you are having the stages in cheptanu. From the explant you will be getting the callus అనేసి అని చెప్తున్నాను. From a single callus you can have about 400 to 500 plants ని మనము డెవలప్ చేయొచ్చు. From a single callus plate you can develop 400 to 500 plants ని మనం చేయొచ్చు. అంటే from a single small disc you will be able to produce so many plants and matter. That is called as a micropropagation. And at the small level, under the laboratory condition, using a very small explant through callus medium, you can develop so many n number of plants ని మనము ప్రొడ్యూస్ చేయొచ్చు. ఇప్పుడు చూస్తున్న బయట నారు కానివ్వండి, ఇప్పుడు చూస్తున్న బయట ప్లాంట్స్ కానివ్వండి, either it can be banana, it can be any plant, వంకాయ కానివ్వండి, so many plants are there. All those plants are tissue cultured plants అన్నమాట. Everything they are tissue cultured plants only. So that's why they are selling it for 1 rupee, 2 rupees, like that they are selling outside. So in this micropropagation, how you can do, and a micropropagation also you will be doing anamata. How you can do so many methods on the first is embryogenesis. And embryogenesis means it looks like embryos, but they are not direct embryos anamata. They are not embryos formed through the fertilization. That you must remember here. So embryogenesis is direct embryogenesis or indirect embryogenesis, that I will tell you. Next, organogenesis, that is organs will come via the callus formation. Or you have direct adventitious and thick adventitious roots etla vastayo, like that you will have the organs formation you will be getting directly. Then you have micro cutting. So you have meristems and the shoot tips or the bud cultures untadi, then micro cuttings chesi kuda you can put in the culture media, you will be getting lot of plants. So first we will see embryogenesis. I told you embryogenesis lo first one is the somatic embryogenesis. So in the local somatic embryogenesis is, mind you, the word here somatic anesari manam chuskoవాలి. Somatic is always the vegetative thing anamata. It is not, you have two types of cells in the body, somatic cells and the germline cells anamata. Germline cells lo meiosis avutundi, that is used for reproduction ki adi use avutundi anamata. Very other whole body, either human beings kanivandi, animal system kanivandi, plant system, remember it is somatic cells only. So here somatic cells lopala you don't have union of pollen and the ovule, manaki fertilization undadu. That you have to remember. Tissue culture you don't have union of the male and the female gametes. Directly you'll be getting the plant system. That's why you call that as somatic embryogenesis. So here you will be having here directly somatic embryos are formed here, or they are non-germ cells. That means they don't come under the reproductive cells, that is the germline cells. So usually they are formed in the callus intermediate stage, callus locally you will get. So there are so many like things, heart-shaped things here. So all these are somatic embryos. So you can see here n number of things. You take them and you put it in the media, so many will come. But ఒక seed తీసుకొని if you put, only one plant will come. But same callus లోపల see, so many somatic embryos are there. So many plants will come here. Okay, so this is called as somatic because there is the formation of embryos are formed from the somatic cells, not from the germline cells. See, somatic embryogenesis. Explant తీసుకోవాలి. From the explant, you put it, you will get the callus. Callus lopalanta like this. So many embryos will come here. See, the embryos will look like this. You take out all the embryos and you put them in the medium. When you put them in the media, you will be getting the plants. మనకి ఇట్లా వస్తుంది అన్నమాట. Okay. So this is in the peanut. Peanut అంటే ground nuts లోపల. సో గ్రౌండ్ నట్ లోపల మనము ఏం చేస్తాం, మనం ఒక్క పల్లి తీసుకుంటే యు గెట్ ఓన్లీ వన్ ప్లాంట్. బట్ హియర్ వాట్ యూఆర్ డూయింగ్, యూఆర్ టేకింగ్ ద సొమాటిక్ ఎంబ్రియోస్ తీసుకొని క్యాలస్ పైన ఈ సొమాటిక్ ఎంబ్రియోస్ ని మనం యూఆర్ సపరేటింగ్ ఇట్ అండ్ యూఆర్ పుట్టింగ్. సో ఇవన్నీ ఉన్నాయో సీ సమ్ 400, 500, ఆల్ దట్ యూ డూ కల్చరింగ్, యూ గెట్ సో మెనీ ప్లాంట్స్ యూ విల్ గెట్ హియర్. నెక్స్ట్ ఇస్ ద ఆర్గానోజెనిసిస్. ఇన్ దిస్ ఆర్గానోజెనిసిస్, యాజ్ ద నేమ్ ఇండికేట్స్, ఆర్గన్స్ ఏ మనకి డైరెక్ట్ గా ఫామ్ అవుతుంది అనమాట. అంటే డైరెక్ట్ గా ఎక్స్ప్లాంట్ ఒక్కొక్కసారి మనకి డైరెక్ట్లీ ఫ్రమ్ ద ఎక్స్ప్లాంట్స్ యూ విల్ బి గెటింగ్ ద రూట్ సిస్టమ్ అండ్ ద షూట్ సిస్టమ్, ఆర్ క్యాలస్ వచ్చి క్యాలస్ నుంచి కూడా యూ విల్ బి గెటింగ్ ద రూట్ అండ్ ద షూట్ సిస్టమ్ అనమాట. బట్ వాట్ ఎవర్ ఇట్ మే బి, యూ విల్ బి హ్యావింగ్ ద ప్రొడక్షన్ ఆఫ్ ద రూట్స్ అండ్ ద షూట్స్ ఆర్ ద లీవ్స్ మనకి డైరెక్ట్ గా వస్తుంది అనమాట. సో అది ఇట్ కెన్ కమ్ డైరెక్ట్లీ ఫ్రమ్ ద what you call explant and pre-existing meristems antam, or without differentiation and explant direct ga it can come, or it can involve embryogenesis through callus also it can occur. Okay, see these are the direct embryogenesis, direct organogenesis. And you take the explant lo you put the suitable hormones. In the suitable hormones you will be getting this particular thing anamata. You will be getting the plants manaki vachesas. First, so here shoots are there. Next you take it, you put it in the rooting hormone, roots will be formed anamata. Okay, so somatic embryogenesis and organogenesis, for both these methods it can be used for the micropropagation manamu use cheyochu anamata. Okay, then this micro cutting. So what is this micropropagation? So micropropagation is nothing, but it is just a science, or it is a way of doing multiplication of plants under in vitro conditions. Adoka art anamata. So based on the science, based on our technology, what we are doing, we are playing with the plants. We are producing multiple plants from a single explant, under the laboratory conditions, which is called as a in vitro conditions. Then usually what you have, usually you will be getting the meristems with or without the callus stage manaki untundi anamata. So from that you will be getting the plants manaki vastundi anamata, as I have showed you earlier. Okay, so what are the steps? So first you have to select the mother plant ni manam select chesukoli. After selecting the mother plant, you have to go for sterilizations. After the sterilization, you put that in the initial growth media. What I told you, initial growth media, both hormones will be equal. That means the auxins and the cytokinins will be under the same ratio. Then next you have to go for multiplication. So for that, what you will do, you will take the explant and first you will be putting it in the shooting media. In the shooting media, the cytokinin will be more when compared to the auxins and matter. Next, you will be going to the third stage, that is the rooting. Until after the shoots are formed, you put them in the rooting media. Then here the roots will be formed. After the roots are formed, what you do? You take this explants and you put it in the small vermiculite, not vermicompost. Vermiculite will be there in the laboratory condition only. After putting that, after its certain growth, you put that in the sterile soil in the laboratory condition only. That is called as hardening. So after that, you will take it and you will put it in the what do you call soil you will be putting, or you will put it in the glass house or the net house you will put. After that only you will be taking it to the field level. See like this. See, you're taking the meristem part, meristem or the shoot tip they are taking. So we have to put the apical meristem in the media. Then from that you will be getting the what do you call shoot system. Then after the shoot system, you put that for the root system, rooting hormone. Then from that you will be getting the whole plant. You transfer it into the sterile plant. Okay. So why is micropropagation important? Enduku micropropagation important? Now it has so many advantages it has. So you from first one is I told you from a single plant from a single seed you're getting a single plant. But for the business purpose or for the development of science and or for the business and what you will do the micropropagation. So from single explant you will get 100 plants. So you take 10x plants, how many plants you'll get? 400 plants you'll be getting. So that 400 plants, if you take at least to the field level, at least 200 plants or 300 plants will survive for us. So that is you're propagating it and you're selling it for the more cost. So that is one basic thing you're having here. Basic thing is for the business purpose and also for the research purpose we can say. But what features it has? Induku manam micropropagation kellali. What features it has? First one is clonal reproduction. So clonal reproduction ante enti ante always when human beings are there or the organs are there, it should maintain heterozygosity. Ante manaki two genes, manaki two sets of manaki diploid. Ante one we will be getting from the mother, one will be getting from the father and mother. So both it becomes heterozygous. Now if suppose mother there rendu saarlu chromosomes vachinayi anukondi, what it will become homozygous anamata. So homozygous lopala the viability will become less and less. The hybrid vigor or the other disease resistance or any other characteristics will increase as the heterozygosity increases. So the clonal reproduction will be maintained by this heterozygosity in the micropropagation. That is one future. Next, in this you will be having many multiple stages you can have. You can recycle it so many times with unlimited clones. Manam chesukochu. So many clones you can do. So mostly what you can do it for the commercially for ornamental plants, లేకపోతే వెజిటేటివ్ ప్రాపగేటెడ్ ప్లాంట్స్. సో మెనీ ఆర్కిడ్స్ ఇప్పుడు బయట వెనకటికి యూ డింట్ గెట్ వన్ ఆర్కిడ్ ఆల్సో. Orchids were very, very rare to see anamata. You never used to get anthuriums. You never used to get gerberas, so many. But nowadays, because of this chamanti ganivandi, chrysanthemum ganivandi, or you have the orchid plants ganivandi, so many plants we are getting, or ornamental plants so many are getting, they are cloning and they are getting micro propagations. Then it's easy to manipulate and productions and ante easily you can do the manipulations anamata. Ante field seasons, environmental influences amanta emi undadamata. You don't see all those things in the micro propagation. Why? Because you have standardized the procedure. According to standardized procedure, you will be doing it in the laboratory conditions. Finally it will go out anamata. So when it goes out, it can maintain hardening ఒకసారి it is done in the laboratory conditions. Outside it can survive very easily. Next, one more thing important here, what you have to do is most of the time micro propagations they will be doing with the apical meristems tissue. Apical meristem tissue lopala you will be having the meristems. Meristems always they are disease free. Meristems always they are disease free anamata. Just because you're culturing, you're doing the whole tissue culture with the shoot meristems or the apical meristems, the plants which are produced will have that meristems characteristics it will have anamata. So naturally when the apical meristem is మనకు అది డిజీజ్ రెసిస్టెంట్ ఉంది, దానికి డిజీజెస్ రావు, వైరస్ ఫ్రీ అండ్ డిజీజ్ ఫ్రీ అనుకోండి. సో naturally all the plants which are produced also will be disease free అన్నమాట. సో disease free plants can be produced. మనకి ఇప్పుడు essay type questions you will be getting అన్నమాట. How to get disease free plants through tissue culture అనేసి. It is through you will be using the shoot meristems or the apical meristems. Through that you will be getting the disease free plants అన్నమాట. Suppose if the meristems లోపల diseases వచ్చినాయి అనుకోండి, ఏమవుతుంది? The plant will not grow at all. The plant will not grow at all. It will not have the capability అన్నమాట. The meristems itself will have rejuvenating cytoplasm. It will have all the enzymes. All it will be having. It is because it's just growing cells కాబట్టి all rejuvenating things will be there. There you will never get in any of the meristems, intercalary meristem కానివ్వండి, axillary meristem కానివ్వండి, apical meristem కానివ్వండి, in the meristems you will not get diseases at all. Okay. Next we have the embryo culture. So embryo culture you can do it by rescuing certain embryos. Okay. Suppose if you have certain plants, from that you take the embryo and you take that directly, you take that embryo and do tissue culturing. So you will be getting the plants you will be getting. So here what you have to do is you have to learn certain things in this. So for certain type of plants, they do the rescuing of the embryo and how I have to explain you, you have polyploidy. In the polyploidy, you have two different types of ploidies or two different distant related plants ఉంటాయి అన్నమాట. So a distant related plants, you do the crossing అన్నమాట. After doing the crossing, you will be getting it. But it may not give the plant, it may not grow into the plant అన్నమాట. So అట్లాంటి వాటిది after the fertilization, when the embryo is formed, you take that embryo, you rescue that embryo. ఆ embryoని నువ్వు కాపాడేసి you take it and you put it in the tissue culture condition. See, this fellow is telling you what they are telling. Embryo culture developed from the need to rescue embryos from the wide crosses where fertilization occurred, but embryo development did not occur. Wide crosses, for example, నేను మీకు మనకి మన ఆర్గానిజమ్స్ లోపలనే చెప్తాను, నేను హ్యూమన్ బీయింగ్స్ లోపల కూడా యు ఆర్ హ్యావింగ్ ఇన్ విట్రో ఫర్టిలైజేషన్స్ అనేసి, ఐవీఎఫ్ బేబీస్ అనేసి అని అంటారు అనమాట. సో వాట్ దే డూ ఆఫ్టర్ ద ఎంబ్రియో ఫార్మేషన్, పిండము తయారైన తర్వాత యూ పుట్ ఇట్ ఇన్ టు ద హ్యూమన్ బీయింగ్ మదర్ వూమ్ లోపల పెడతారు అనమాట. సేమ్ లైక్ దిస్ వైడ్ క్రాసెస్ ఉంటుంది, అంటే అన్రిలేటెడ్ ప్లాంట్స్ విత్ క్రాసెస్ ఉంటాయి అనమాట. సో ఇన్ దోస్ ఆర్ related only, ఒకటి wild variety or one is domesticated variety. If the cross is done in between both of them, you will get the embryo formation, but that embryos will not grow into the plant anamata. So padipodamo, leaf develop gaakapodamo, something or the other it will get dried up. Atlanti what is, after the embryo is formed, you rescue it and you take it to the tissue culture and you will get the new plant anamata. Next, this embryo culture can further be developed for production of plants from embryos developed by non-sexual methods. What is this non-sexual methods? I told you, tissue culture itself is somatic cells. అంటే మనకి somatic cells లో మాత్రమే, vegetative cells లో మాత్రమే మనకి జరుగుతుంది అనమాట. మనకి you don't have జర్మ్ లైన్ సిస్టమ్ ఇక్కడ మనకి ఇన్వాల్వే కాదు, మియోసిస్ ఇక్కడ మనకి ఇన్వాల్వే కాదు. హియర్ యు మస్ట్ కీప్ దట్ ఇన్ మైండ్. ఓకే. సో హియర్ వాట్ దే ఆర్ టెలింగ్ ఫ్రమ్ నాన్ సెక్షువల్ మెథడ్స్. ఇప్పుడు హ్యాప్లాయిడ్ ప్లాంట్స్ ఉంది, పోలన్ గ్రెయిన్ తీసుకోండి, ఓవ్యూల్ తీసుకోండి. యు ఆర్ హ్యావింగ్ ఓవ్యూల్ కల్చర్, యు హ్యావ్ యాంథర్ కల్చర్, యు హ్యావ్ పోలన్ కల్చర్. Anther is what? Anther or pollen grain is what? It's a male gamete. That what you are doing, doing the tissue culture. It is only single. It will have only, it is gametes cavity, it will have only one set of chromosomes only it will have. Ovule undi. Ovule lopala already you have the meiosis అయిపోయి ఉంటది. శాఖన విభజన అయిపోయి ఉంటుంది. సో దాంట్లో మనము ఇఫ్ యూ డెవలప్ ఫ్రమ్ దట్, అందులో కూడా యూ విల్ బి హ్యావింగ్ ఓన్లీ హ్యాప్లాయిడ్ సెట్ ఆఫ్ క్రోమోజోమ్స్ మాత్రమే మనకి ఉంటుందన్నమాట. ఏకస్థితికమే మనకి ఉంటుందన్నమాట. సో అట్లాంటి హ్యాప్లాయిడ్ ప్లాంట్స్ యూ డూ డబ్లింగ్, అండ్ ఆఫ్టర్ డబ్లింగ్ ఇట్ యూ టేక్ దట్ ఫ్రమ్ దట్, you take that and you do the tissue culture anamata. So you will be getting the new plants, you will be getting anamata. Okay, so same ade, you take the embryo, you put that in the medium, from that you will be getting the plants manaki vastund anamata. Okay, next haploid productions. I told you embryo rescue, you will be getting the what do you call haploid plants, or anther culture. Through the anther culture or ovule culture, you will be getting haploid plants only, because anthers are the male gametes, they represent the male client, and the ovules, they represent the female client. Andukani there you will be getting haploid plants only, because after meiosis శేఖరణ విభజన తర్వాత మనకి అది ఏకస్థితికం అయిపోతుంది అనమాట. మళ్ళీ ద్వైస్థితికం కావాలి అంటే it requires fertilization is required. So here no fertilization is required, but from anther culture also you will be getting plants. Our plants, you call that as haploid plants anamata. Ovule also, from the ovule culture also you will be getting the haploid plants. You will be getting plants will be produced, but seeds will not be produced after doubling the chromosomes also. So here you will have the advantage of producing seedless variety grapes, seedless variety of watermelons. Itlantivi, all these can be exploited from this haploid productions. So we will see this bulbosum method of this haploid production. So how they have rescued the embryo. So they have taken two things. One is Hordeum vulgare, and this is barley anamata. So one is a wild variety. Ante manamu adividi wild variety ante adividi first variety edaithe undo adavi jatidi, it is that wild variety. So దాని తర్వాత now we are domesticating కదా మనము అంతా. సో we have domesticated అవి మనం we are eating that అనమాట. So the wild variety is Hordeum bulbosum and this is Hordeum vulgare. So they have done crossing it between these two చేస్తారన్నమాట. So when they crossed in between these two, naturally fertilization అంటే pollen and ovule fertilization has taken place, and you had embryo formation అయింది. But the embryo was not growing anamata. The embryo was not giving a new plantlet anamata. So what they have done, they have done the embryo rescue చేస్తారన్నమాట. So in this embryo rescue, what they have done is, these both are 2n, but here they have eliminated the chromosomes of this wild variety. They have eliminated this chromosomes of this wild variety. Hence 14 has become 7. And then what they have done is, they have done the doubling. They have done chromosome doubling they have done, and they have grown this plant, but they have created a haploid barley plant. They have created anamata. So from that time you call this method as bulbosum method. So eliminating one set of chromosomes and rescuing the embryo, and you get the plant. So that method they have called as bulbosum method. Okay, then next it can be anther or microspore. It is a male gamete. Okay, so same, you take the pollen grain. So this is the anther. So anthers is these, you directly culturing the nutrient media. So it is two types.